going to start talking about the visual system. We actually will have 2 whole exam sections on vision, pretty much focusing on bottom-up concepts for the whole first exam, and then we'll look at top-down influences for the second exam. That's how much we know about vision. Remember I said we know the least about the chemical senses and touch? We know so much about vision, about the intricacies of how it works, and it's so complex that there's just a lot more content out there. Probably 2/3 of your textbook is on vision, right? And so then the last section we'll talk about the auditory system. So, what do we start with if we're trying to learn a sensory system? Yeah, what is the stimulus and how does it get transduced? What are the receptors, right? So we're going to start bottom up and go from there. So the stimulus for the visual system is light, but really what light is electromagnetic radiation. There's a lot of electromagnetic radiation out there. We only detect, we only see a very narrow range of electromagnetic radiation. So when we say, what is the stimulus for the visual system, do you think that might be on an exam? Probably, yeah. So when we say, what is the stimulus for the visual system, it's 380 to 760 nanometers of electromagnetic radiation. That's what it is, right? And so, and that 380 to 760 wavelengths corresponds to the quality of the stimulus. So the shortest wavelengths, 380, are blue, and then we go through your little Voyagey Biz thing, right? So blue, green, yellow, orange, red. Right? All right. So the quality, the hue, the color is associated with the wavelength of electromagnetic radiation. We also then have intensity. So when we talk about thresholds, we have a range of sensitivity, and we can't detect 350 nanometers of light. We can't detect 800 infrared wavelengths of light, of electromagnetic radiation. You can only detect in that narrow range. So above and below is above and below our threshold of the sensitivity. We also then have intensity above and below. Right? So how bright does the light need to be in order for us to detect it? And so there are 2 parts that when we talk about brightness, we have illuminance, which is the amount of light that hits a surface. So if we talk about that bulb in that projector sending white light in my face, that is illuminance, the number of lumens, the amount of intensity of light coming off of that bulb. And that bulb's rated for something, right? So if we talk about a light bulb in your room, it might be 60 watts. That's going to produce a certain amount of lumens. Like now with LED bulbs, they really are going with lumens, and they sort of tell you how much light it puts off instead of how much energy it uses. So 60 watts is how much energy a light bulb uses, but it's putting off a different amount of luminance. Then luminance is the amount of light reflected from a surface. So this surface, this whiteboard, is created to maximize reflecting light off of it because it's a screen, essentially, right? And if I stand in front of it, right, my shirt, even though it's a light-colored shirt, is not having as much luminance come off of it, as much light reflected back to you, right? So because you're not standing right in front of the light, you're not seeing any illuminance. All of this is reflected light back to your eyes, which would be luminance. Good? All right. Radiance is the amount of light produced by the source. Radiance is the amount of light produced by the source. So here, the amount of light being produced by that bulb and the amount of light that's hitting the surface are pretty much identical. There's probably not any decrease in the radiance to the luminance, right? But you can think about if we had a regular 60-watt light in here and the lights were off, that it's giving off radiance, and the surfaces around here are going to have an illuminance that's greater than the back wall. The light's going to degrade as it reaches back there. It'll be darker back there. It won't have as much illuminance. So that's the difference between radiance and illuminance. Okay. All right, so we're good on physical properties of electromagnetic radiation. We are all good. Great. All right, so we need to talk about the eye because that's where the receptors for light is. And so the eyeball is full of fluid. There's no air in there. And so we have a cornea and a lens that are going to be involved in helping us focus light waves on the rear of the eyeball, which is where the retina is. And in between the cornea and the lens is fluid called aqueous humor. And in between the lens and inside the eyeball is what we call vitreous humor. Two different types of hopefully clear, because if it's not clear, if it becomes cloudy, then it's going to interfere with the light waves, right? So clear fluid that are in there. Yes?
Wait, so like, if you do like over your eyeball, you like see something like that. I realized that they were like kind of hollow, like, not hollow, but like,
Right.
it's not tissue in there.
No, it's hollow and it's got fluid in there. And so there's the black dot, right? The black dot in the middle of your eye is black because light's going in and being absorbed and not coming back out, right? It's just a hole called your pupil. Right? And then your iris is the colored area around your pupil, and that your iris then can adjust to allow more or less light to come in. So we talk about our pupil being constricted and small or dilated and big, right? And that's a change in the size of the iris that's doing that. The iris is actually like a muscle that can open and close. All right. So we need to understand a little bit about properties of light. But we have the ability to bend light and magnify light and focus light. And so refraction is where the cornea does refraction, and it actually takes light coming in and focuses it to increase the power. It's almost like a magnifying glass, if you will. And so the cornea does about 3/4, the majority of the refraction of the eye. And then the lens does the rest. And the lens not only inverts the image on our retina, so everything is backwards and upside down when it's projected to our retina, but it also focuses that image on our retina so that it's not blurry. All right. So that process of the lens focusing the image on our retina is called accommodation. Accommodation. That means the lens is either elongated or contracted in order to focus the image. Right? Now, the length of your eyeball, the distance between the lens and the retina, is usually matched to the refractive power of the cornea and the lens. But in the case where it's not, either the eyeball is a little bit more like a football shape, a little more elongated, or a little bit smushed where it's not exactly round, right? Then we get conditions like myopia, where because the eyeball is a little bit longer, so in a normal eye, healthy eye, that image comes and gets focused right on the retina. But in an eye that's too long, that point of focus happens a little bit before the retina, and so by the time the image gets to the retina, it's slightly out of focus. That's myopia. And hyperopia is the opposite down here, where the eyeball's a little short, and so the image is not yet reaching that focal point before it gets projected onto the retina. These are also called farsightedness and nearsightedness, myopia and hyperopia. And those are easily corrected with the lens. So you get glasses that have either a convex or concave shape. The concave or convex shape is correcting either moving it forward or pushing it backward, right? Pushing it backward in the case of myopia, moving it forward in the case of hyperopia, as a corrective lens to make sure that it's focused there. And so whatever your prescription is based on how much movement has to happen to get that put back on. Contact lenses, same thing, it's just we've taken that and sculpted a little piece of plastic that goes on your eye that's going to do the same corrective thing. Okay. All right. So when objects are far away, about 20 feet or more, or further, our lens is totally relaxed. And when our lens is totally relaxed, everything being normal, meaning no myopia or hyperopia, then that is just projected regularly in focus on our retina. Okay? But as things get closer to us, the image becomes unfocused. And in order to move that focal point from back here back onto the retina, our lens has to contract and get fatter. And when the lens contracts and gets fatter, it moves that image onto and focuses it on the retina. So we can do that right now. So if you hold up your finger in front of me like this, okay? And then look at me, okay? And then look at your finger, and then look at me, and then look at your finger, right? You should be able to kind of feel the difference. Not only does your lens contract, but your eyes also have to converge in order to keep. All right. So as you get older, towards my age, those muscles that help contract the lens start to get weak, right? And so when you go out to dinner with your parents, right, and they read the menu, what do?
Turn the light on. Or did it like squint up?
Or they hold it like this, right? They try and hold the menu as far away as possible because the further away it is, the less they have to contract that lens. If things are up really close, they really have to contract that lens, and it's difficult to do that because those muscles get weak after a time. So what else do they do besides hold the menu way out here? They put on their readers, right? They put on their readers. So these are just simply doing this magnification. It's a glass set to help us focus on short, close-up things, right? And they only need it when they're reading, when they're looking at things up close, right? And all of you will probably at some point in time need readers because your muscles just weaken over time and you're not able to contract as much.
So what's it called when you have to like squint to see stuff? Like if your eyes are really bad? I think I may not have found it myself. Like, is it like it's hard to like
squinting is maybe you're trying to focus on something, you're trying to make it contract or something like that, but you're not doing it.
Okay.
Okay, so we're finally getting to where receptors are, which are in the retina. Okay, and so in the retina, back here, if we take a slice of the retina and we blow it up and look at these things, we see that there's a center of vision. So when things are directly in front of us, they're being projected on a spot of the retina called the fovea. So the retina is mapped to our world. So things that are up here in the world, the ceiling, are being projected down here on the bottom part of our retina. And the floor of the carpet is being projected up here. And so there's a map from floor, center of vision, ceiling, all the way around the retina, and that doesn't change. That doesn't change. So we have receptors back here, and then we have an optic nerve that is going to take the output of the receptors and run it to the and run it to the brain, right? To the LGN, to the thalamus. So, yes?
So you're saying that like the ceiling would go towards the bottom?
Yeah, because everything is backwards and upside down, right? So here's our coffee cup, and the coffee cup, the top of the cup is at the bottom, and the saucer's at the top. So already we're getting confusing with things, right? Because we've already flipped our whole world upside down and backwards.
Has there ever been like, this always sounds so dumb, but has there ever been a case where like something's gone wrong and someone is actually
Yeah, we'll talk about that.
terrifying?
Not gone wrong, but where we just cut the nerve and take the eye out and flip it over and put it back in, and then everything's reversed. So we'll talk about that. We'll talk about that experiment. Not today, but we will talk about that. All right. So here, what are the receptors for the vision system? You guys know? Rods and cones. And why are they called rods and cones? Because they look like rods and cones. Right? It's one of those easy things, right? And so we have rods and cones here, and the anatomy of the eye is kind of funny because we have light coming in, and the light actually shines through these layers of cells back here, and the rods and cones are positioned at the back of the retina, right against this stuff called the pigment epithelium. And then they activate the rods and cones. The rods and cones then activate a group of what we're going to call processing neurons. Processing neurons here, bipolar, amacrine, and horizontal cells. And then those cells, what do you think those cells do? Process the signal, exactly. So the signal that comes in the optic nerve, so processing cells, they're hooked up to the optic nerve. We call these ganglion cells. So the signal in the ganglion cell going to your brain is already not what's actually out there in the real world. It's already been manipulated and processed by these cells in your retina before it ever even gets to your brain. So going back to the very first day of class, when Plato said don't trust your senses because they're not really telling you what's out there, in vision, that really is the case. You are not seeing a 1-to-1 ratio of light coming in and the signal going to the brain. It's already been modified by the time it starts going to the brain. Is that crazy? Not only that, your visual system is constantly making up information. Constantly making up information about what's out there in the world. And the reason it has to do that is because because this is a backward system, right? All the ganglion cells are on the inside of the eyeball, and they have to get out. So they all come together in this bundle called the optic nerve. It's called the optic disc. And what do you notice is missing where the nerves exit? What's missing right here? Rods and cones. So what does that mean? You have a blind spot. You cannot see right there. Right? You cannot see right there. So take one of these and pass it down. And then not everybody's here. We'll just got groups and fours. Take one of those and pass it down. I'm going to prove to you that you have a blind spot. You have 2 blind spots floating around out there, and your brain is making up information. So if you cover your left eye, okay, and then with your right eye, focus on the cat and hold the paper about this far away. You kind of move it back and forth, and you don't move your eye off the cat, and you make the dog disappear. Like right now, I'm looking at the cat, and there's no dog in the cage. Can you do that?
I can just see its tail.
Right? You got to move it real slow, and what you're doing is you're essentially putting the dog in your blind spot. You're putting the dog in your blind spot, and when you put the dog in your blind spot, what happens to the cage? It's empty. It's not that there's a black spot there. You actually see lines going straight through, and there's no dog. Because your brain is constantly looking at the receptors all around the optic nerve, all around the optic disc, and it's making up, it's filling in this missing information based on what the surrounding is doing. So when you put that dog right in the middle of your blind spot, the brain's like has no cue to put a dog there. It just sees the lines around the surrounding and runs the vertical lines straight through. And so the cage actually becomes empty. Alright? Alright, so your brain is constantly lying to you. You have these two big blind spots out there. We don't see two black dots because our brain's just making up information and filling it in the best that it can. And unless we do something like put something directly in that blind spot so that there's no other cue available to the brain, it does a pretty good job. But if we do that, we really become blind to that thing. All right. So this is a actual microscope picture of the retina, and we see these layers. So we have this thing called the pigment epithelium here, and then we have the photoreceptor layer. So this is the rods and cones. Rods and cones are broken into outer segments and inner segments, and then they hook up to this layer of processing cells in which we have bipolar cells, horizontal cells, which have connections laterally, amacrine cells, which have connections laterally, bipolar are connecting from the receptor to the ganglion cell, and then we have our ganglion cells that are going to exit and become the optic nerve. The axons of the ganglion cell are the optic nerve. So why is this all backwards like this? Why does the light come in like this? Well, it turns out that these rods and cones use a lot of energy in order to transduce light. And because their energy needs are so high, they have to be back here next to the pigment epithelium that can supply nutrients and to them. So if you think about it, if it was flipped around, and the rods and cones were out here sticking into the eye, and then the signal went back, how would we get nutrients to them if they're hanging out in the vitreous fluid out there? Right? It'd be very difficult. So that's why it's organized backwards, so that these rods and cones have access to the pigment epithelium, which is going to provide that nutrient support. The other thing the pigment epithelium does is any photons of light, any electromagnetic radiation that's not detected by the rods and cones gets absorbed by the pigment epithelium, and therefore it doesn't bounce around or cause reflections or anything like that. All right. So again, rods and cones, receptors, a layer of processing cells, bipolar, horizontal, and amacrine. And then we have our ganglion cells whose axons are the optic nerve. It's going to go from your retina all the way into the lateral geniculate nucleus of the thalamus. All right, so rods and cones. Differences between rods and cones besides their shape. Okay? Their shape is different in this thing called the outer segment. The outer segment is where the photopigment is where the light is detected. The inner segment has the nucleus and other sort of machinery of the cell. And then we have synaptic contact with the processing cells at the other side. Rods designed for low intensity light, and they cannot do color discrimination. So how do you know if you're using your rods or your cones? Even if it's dark, how do you know if you're using your rods or cones? If you see color, you're using your cones. If you see grays, different shades of the same color of gray, you know you're using your rods, right? So have you ever been in your dorm room when it's really dark and you're trying not to wake up your roommate and you're going through your t-shirts and you can't tell, you can see like dark colored and light colored t-shirts, but you can't tell what colors they are, you're using your rods.
So Milda's sister, she can only see black and white. So like, does that mean she only has rods?
No. It means she only has one type of photopigment in her cones. Cones see high intensity light. If she only had rods, she'd be blind unless she was in dark, unless she's in dark, dim light, right? Because rods become useless in bright light. Rods only work in low light levels. Cones have to have high intensity light to activate them. They don't work in low intensity light, but they can detect color.
Is there like a threshold for the intensity of light that like
There is, and next Monday you're going to detect it in lab. All right. You guessed the lab. You win. All right. If we look at the placement of rods and cones on the retina, they're not evenly distributed. They're not evenly distributed. The fovea, 0 degrees, that's our center of vision, is our best discrimination. And we have the highest amount of cones there. So here's our fovea, 0, right? What's going on over here? Right, that's where the optic nerve is, right? So there's 0 receptors, 0 rods, 0 cones in that area right there. About 20 degrees off center. Okay, so if we actually look, I'm going to show you another picture where we're looking at the same distribution of rods and cones, and we're looking at actual photomicrographs of that. We see right here at the fovea, we have so many cones packed into our fovea that there's essentially no room for rods. So the rods drop off, and we really have no rods right in the center. Rods are bigger, you can see them here, the cones are packed in and around the rods. Right? And so, conversely, out here in the periphery, we have less cones. Right? Limited resources, we can only fit so much stuff in there. We concentrate the cones in our center of vision. That gives us our best color, detail, discrimination, sensitivity ability right in the center of our vision. Right? And then less out here. So what does that mean if you're in very dim light and you're using your rods? So you're in very dim light using your rods, cones aren't active. What does that mean? Yeah, the periphery is actually going to have better detection. It's something right in your center of vision is going to be really hard to see because you don't have very many, if any, rods right in the center of your vision.
Huh. Is that why at night it's so much easier to see out of your peripheral vision?
Yes.
Interesting.
Yeah.
I just thought it was me.
Yeah, no. There you go.
I can't listen
It's
to
your
you.
anatomy. It is you. It's your anatomy. All right. So rods, there's one type of photopigment in rods. It's called rhodopsin. One type of photopigment. And that photopigment responds maximally to sort of blue-green light and then drops off on either side of that, and it's harder to stimulate that, right? One photopigment has a tuning curve, responds maximally to red to blue-green light, and then it drops off. Actually, rods cannot detect pure red light. So pure red light cannot be detected by rods. Okay? All right. We'll talk about why that's important. Hopefully we get there. There are 3 types of photopigments in the cones. So how many receptors are there for the visual system? 2, rods and cones. How many types of photopigment are there? 4. 1 for rods, 3 for cones. Okay? So there's not 4 receptors. There's 2 receptors, rods and cones, but there's 4 different photopigments. Rhodopsin for rods, and then 3 different ones that make the cones essentially into 3 populations of receptors. Ones that detect short wavelength light, blues. Ones that are tuned to best detect greens, medium wavelength. And ones that are tuned to best detect reds, long wavelength cones. So we have short, medium, and long wavelength cones. And it's the ability of these cones to detect different wavelengths that allows us to see color. Because we can now look at the output of those 3 different receptors and tell what color is present. So in blue light, if we're looking at blue light, what cones are active? The short and not the medium or long wavelength, right? If we're looking at the red chairs in this room, what's being activated? How do we know it's red, not blue? Because long wavelength are being activated and short are not. With rods, how many photopigments do we have? 1, right? So that's why we can't detect color, because we don't have anything to compare it to. The Only thing we can see are intensity. High intensity, brighter shade of gray, low intensity, darker shade of gray. Y'all get that? Okay. All right. So, certain wavelengths of light differentially activate the short, medium, and long wavelengths, and these are kind of striped because yellow is where? Right in the middle of green and red, so yellow equally activates medium and long wavelength cones. So if we're looking at my shirt and I'm saying that it's yellow, right, then what is happening is your medium and long wavelength have equal outputs, and the brain interprets equal outputs as the color yellow. What happens if we have equal outputs from short, medium, and long wavelength? All three have the equal output. White, right? The absence of color, white light. If we have no output from all three, what is it? Black, right? The absence of light, no color. So this is why somebody who can only see in black and white or shades of gray is what we call monochromatically colorblind, only one color, and they don't have three photopigments, they just have one, right? And they're able, it's like having rods, but for a high intensity light. Right? That's a very unusual type of color blindness. Most color blindness is that you have two of the three, you're only missing one of the photopigments.
Is it genetic?
It can be.
Because her dad has it.
Yeah. And it also is a sex linked to the X chromosome, so it's much harder for females than for males. All right. And we'll talk about that when we talk about color deficits. We'll talk more about color. All right. So how does this we still haven't gotten to transduction before. Still on results. We're going to look at rods just because they have one photopigment, and we'll just use them as a model. Same exact thing is happening in short, medium, and long wavelength, but it has to be short wavelength light, medium wavelength light, or long wavelength light to activate those. In rods, so here's our rhodopsin molecule. It's found in, there's these plates, these discs that are stacked up in the outer segment, and they're full of photopigment, rhodopsin. And when light comes in, it activates, this is almost like, kind of like a metatropic receptor, but it's not, there's not a binding. The light actually causes a reaction in this membrane, in this photopigment chemical, rhodopsin, that's going in and out of the cell membrane. And when the light hits it and changes that, it releases a signal. A signal gets broken off and released, not unlike a G protein receptor. A G protein gets released, it's not, but it's kind of similar, right? Light comes in, activates that. That signal then actually goes and closes the sodium channel. So here's where it gets complicated. I'm not going to lie to you, this is complex, not simple. You're just going to have to wrap your head around it, okay? So in the dark, that means no light is present. Right? In the dark. There's a sodium channel that's open, and that sodium is flowing in and depolarizing the rod. That means it's getting positive, right? And we call that dark current, because in the dark, there's current coming in, and this rod and cone, when it's not being stimulated by light, is actually releasing an inhibitory neurotransmitter, preventing action potentials from going to your brain.
When it's dark.
When it's dark. And if we don't allow action potentials to go to your brain, then we don't see anything, because we're in the dark. Okay? What is complicated? Transduction is a three-step process. Step one, light comes in and gets absorbed by the photopigment and releases a signal. That signal closes the sodium channel. And when the sodium channel closes, what happens to the inside of the receptor? it was positive here, we close it, and we're not letting sodium in anymore, so what's going to happen? It's going to drop, and it's going to stop releasing its inhibitory neurotransmitter. And if we stop inhibiting signals to the brain, and a signal goes to the brain, what do we do? We see something. Light was detected. Okay? And so that's when rods and cones are activated, they stop inhibiting, and that allows those processing cells to send action potentials to the brain. We call this whole process removal of inhibition. Removal of inhibition. When we remove the inhibition because light was present, action potentials go to the brain and we see. We see. All right. So here's another example. I think this is from your book. And so in the dark, no light's present, the sodium channel's open, dark current, I read that, that's not in your book, right? So dark current's coming in, and it's causing inhibitory neurotransmitter to be released, and we're preventing signals from going to the brain. Light activates rhodopsin, releases a signal, an enzyme, a signal that closes the sodium channel, and then we stop inhibiting, we remove the inhibition, a signal that we detected light goes to the brain. We good? Okay. Let me complicate it just a little bit more for you. Okay. What is an inhibitory neurotransmitter here? Glutamate. What do we talk about glutamate being? Excitatory. Everywhere in your body except in your retina, it is inhibitory. What makes a neurotransmitter excitatory or inhibitory?
The site that it attaches to.
The site it attaches to. So in the retina,
Wait, did you say everywhere in the body except the retina it's inhibitory?
it's excitatory.
Oh, okay.
Right? There's one type of class of glutamate receptors that when glutamate binds to them, they're inhibitory, and it's only found in our retina. Isn't that crazy? All right. So, why do we have, well, first of all, why is this crazy system set up this way? I mean, why wouldn't we just want light to come in and cause neurotransmitter to be released? Why would we want to have it backwards?
Because we're using our eyes a lot more in the light than the dark.
Yeah, so what percent of the day do you have vision? When do you have vision? When do you not have vision? When your eyes are closed, right? And when are your eyes closed? When you're sleeping, right? So for normal people, that's about a third of your day that you're sleeping if you have a good sleep circadian rhythm schedule, right? So for a third of your day, you're having to release inhibitory neurotransmitter. That's costly, right? To release all that neurotransmitter. A lot of energy use, right? But in light, we actually can decrease the neurotransmitter release in the 2/3 of the day that we're actually using this system. So it's actually a very efficient way for the eyes to detect light, albeit very complicated and complex to understand. All right, so why do we have cones and rods? Why do we have these two separate kinds of systems? Discrimination? Yeah, so, yeah, cones are good for color. What kind of world do we live in terms of light stimuli?
There's a lot of it.
Hmm? So when you walk outside on a sunny day, at noon, how bright is it? Pretty bright. And if you walk outside on a moonless night, how dark is it? Pretty dark, right? The range of light intensity in our natural world, sun shining brightly, no moon on a dark night, that range is so huge that one receptor can't be sensitive enough to work in the darkest of nights and not overwhelmed by the sun at noon, right? So we actually have two whole separate systems, a night vision system and a day vision system, in order to accommodate the fact that that range of intensity is so huge, All right, so photopic vision is cone vision, color vision, bright light. Scotopic vision is when we're only using our rods. You know you're only using your rods because you can't discriminate between colors. You're only seeing in shades of gray, right? We have to adapt between these two types of vision. So dark adaptation is the process of going from bright light into a dim light setting. So you walk into a movie theater, go to a matinee, it's bright sunlight outside, you go in and you're trying to find a seat, and is it easy or hard to see where people are sitting and what seats are available? It's hard at first, and then you go find a seat, you're sitting there, and what happens? You dark adapt, and your dark vision becomes better, and all of a sudden you can look around and actually see where people are sitting, right? Then, after the movie's over, have you ever walked directly out of the movie theater out one of those like side exit doors into the parking lot? What's it like? There you have to squint. There you have to, it's painful, right? The process of going from dark adaptation to light adaptation is very painful, right? And so in bright light, rods are, all the photopigment in rods is used up. It's what we call bleached. Bright light, it's so sensitive to dim light that in bright light, it's all vanished. It's just, boom, stimulated, overstimulated, gone. When we go into a dark setting and we're no longer stimulating with bright light in the movie theater, we start making more rhodopsin. And the rhodopsin starts to build up, and the more photopigment we make, the better our dark vision becomes. Now we've been in a movie theater for 2 hours, we walk outside, and our rods, those outer segments of our rods are full of rhodopsin. We walk into the bright light, and all of that rhodopsin is immediately, in 1 second, stimulated and used up, and it's blinding. The signal out of our eyes is so great from the rods that it's painful. Remember, anytime we have a very large sensory signal, our brain interprets that as pain. And so you actually can feel pain when you have light adaptation, right, going into there. Yeah?
Is it just a myth that blue eyes adapt slower?
The what?
Blue eyes adapt slower. I've always heard that.
Blue eyes adapt slower. No, I don't think the color of your iris does anything to do with that. So as we sit in the darkness, right, we get better and better threshold. Our threshold goes down, right? Because we're building up more rhodopsin. Okay? So here we have a rod cone break. So here we're going down in intensity of light, cone, cone, cone. We get to a threshold, now cones can no longer be stimulated. Here we're only in rods. So we have this sort of fast adaptation to this point. And once we get there, we have to wait for the rhodopsin to continue to build up until we have maximum rhodopsin in all of those discs in our outer layers, in our outer segment, and then we reach our maximum sensitivity. And that process takes about 15 or 20 minutes to happen. And why do you think it takes about 15 or 20 minutes to happen? Why do you not think it takes an hour or 2 hours? In our natural world, how do we go from needing to use our cones to needing to use our rods? The sun sets, right? And so that same amount of time to go from bright light to dim light, 15 to 20 minutes, is linked up to here. Our system evolved to be able to handle that. So that as the sun sets, our dark vision comes on board and we're not blind. All right. So rods are bleached or useless in bright light. So all the photopigment in bright light stimulates all this. And so every time a rod makes a new thing of rhodopsin, it gets zapped immediately in bright light. So there's zero available rhodopsin. Every time it makes it, it gets zapped. It's only when we walk in the movie theater that it gets made and then stays and doesn't get zapped that we can build up levels. All right? So rhodopsin's made from two chemicals, retinol and opsin. And so we have this process of rhodopsin. When light hits it, it activates it. The signal gets released. Now it's useless. We have to then rebuild it with vitamin A and opsin. They come together and make a new rhodopsin molecule that's available now for light to hit it and be bleached again. And this is where the myth of like eating carrots gives you better eyesight, because carrots have a lot of vitamin A, right? If you eat a lot of carrots, it's not going to give you super eyesight, right? If you have a diet deficient of vitamin A, yes, it could cause you to go blind. It could cause you to have visual problems because you wouldn't have enough vitamin A available to make this stuff. But you can't have more. You can have adequate, but you can't have more. Yes.
I'm a little confused. So like in the dark, you're saying it regenerates. Is that just it like building up inside, and then it's like all released? You're saying, or what?
Yes. So if we have 20 discs in bright light, all 20 discs have zero rhodopsin. Anytime we make more, the light immediately, as soon as it's made, activates it and it goes away. So we're at zero level of rhodopsin. Even though it's being made, every time it's made, it's bleached, right? So when you go in the dark, we make a rhodopsin molecule, it doesn't get bleached because we're in the dark, and so we're starting to fill those 20 discs full. And so when we get to 100%, we have our best dark adaptation, our best vision. But then when we walk outside, all 100% get bleached immediately. That's this huge signal that goes to our brain, and it's very painful.
Is that why, like, as you step outside, you see like light?
Yes, yes. Because this huge signal goes to our occipital lobe, and we're blinded until, and you can tell when is all the rhodopsin used up, when that large signal goes away. Because now it can no longer send the signal. So it takes a second or two to use up all that rhodopsin. All right. That's where we're going to stop. Make sure that you get visual transduction sort of cemented in your head this weekend. Don't forget labs are due 8 o'clock on Sunday.